Hi, I'm Brian Grant, and you're listening to On Time, the Brian Grant Foundation podcast about living with Parkinson's. If you want to know more about Brian Grant Foundation, go to briangrant.org.
Hi, this is Katrina Call, Executive Director of the Brian Grant Foundation. Thank you for listening to our podcast, which is one of our many programs to help empower people impacted by Parkinson's disease to lead active and fulfilling lives. Our programs are possible because of the generous support of our donors. Please consider helping us continue this work by making a donation at briangrant.org. Thanks again for listening. For many years, researchers have been working to develop tools to help diagnose Parkinson's disease. Today, those efforts are starting to pay off. I'm Katrina Call, the Executive Director of the Brian Grant Foundation, and in this podcast, I'm speaking with Dr. Todd Levine, the Chief Medical Officer and co-founder of CND Life Sciences, about the company's SYN1 test that helps to diagnose synucleinopathies, including Parkinson's. Welcome, Dr. Levine.
Thanks for having me.
So tell us a little bit about yourself and your role at CND Life Sciences.
Sure. Depending how far back you want to go, but I did medical school at Duke, and then did a neurology residency and then a fellowship at Washington University in St. Louis. The particular fellowship that I did was actually in nerve and muscle disease, so it was not in movement disorders. But that was about 30 years ago, and that was at a time when people began to realize that you could study nerves by looking in the skin. So obviously we have nerves throughout our whole body, but they're very accessible just by taking a very small piece of skin. And so that technology, the ability to just see the nerves in the surface of the skin, developed about 20 to 30 years ago. And so I began to incorporate that into my practice as a way to help diagnose nerve conditions. Then over the past decade, so beginning about 10 to 15 years ago, a number of really smart researchers realized that you could not just look at the nerves, but you could actually look inside of the nerves. And you could look and see if there are certain proteins that are building up inside the nerves. And so that was something that we discovered about a decade ago as it relates to alpha-synuclein. And so I got very interested in trying to see if we could then take that and bring that to people who were dealing with the diagnostic dilemmas that often comes with a diagnosis of Parkinson's disease. So we founded CND Life Sciences 7 years ago. We spent the first 3 years kind of perfecting the technique. The technology that we use comes from Harvard and Beth Israel in Boston. And so my two other co-founders are both professors of neurology at Harvard. And then after those 3 years of really kind of perfecting the technique, we began commercial testing, so offering it to patients and doctors around the country about 4 years ago.
That's great. So today the SYN1 test is now commercially available.
Yes.
It's available to patients. How does it test for Parkinson's disease?
So it doesn't test specifically for Parkinson's disease. As your introduction sort of said, it looks for what we call a synucleinopathy. And the reason for that word is that we all have alpha-synuclein in our nerves. And we don't actually know the function of the protein, which is very interesting. But we know it's very critical to the ability of the nerves to work. When that protein exists at its normal state, it sort of is easily dissolved in the water that makes up our bodies, and it just kind of floats throughout the nerves. Something can happen, and we don't know what that thing is, but something can happen that will cause that alpha-synuclein to start to accumulate in clumps. And we can identify the difference between the normal alpha-synuclein and the abnormal alpha-synuclein by doing a type of stain, what we call an immunostain. So we take an antibody that's very specific for the abnormal protein. And we can look inside the nerves and see if this abnormal protein is accumulating. Now, depending on what the patient's clinical symptoms are, that then helps with the diagnosis. So if you have a patient who has a rest tremor and they shuffle and they have this abnormal protein, that it's going to be very likely that that's related to Parkinson's disease. Another very common disorder that is associated with this protein building up is called dementia with Lewy bodies. That's the disease that Robin Williams passed away from. So if somebody was having memory problems and hallucinations and they had this abnormal protein, then the doctor would say, I think this is more likely to be dementia with Lewy bodies.
That is a really interesting explanation of the test. And thank you so much for diving into the science. And while we're on the topic of the test, I know you just recently published a paper showing the test is highly specific, highly sensitive. So for our lay audience, what do those two terms mean?
Great question. So we and others had published small numbers of patients showing that this technique actually worked. But to really kind of validate that, you have to take that to a large number of patients. And then you also want to take it to different types of groups. So we had 30 different centers across the U.S. Some of those were academic centers, movement disorder specialists, and some of those were general neurologists who just happened to see a lot of movement disorder patients. And so we asked those doctors to give us their best clinical diagnosis about what they thought their patient had. What we did then was to take the clinical information, and we had an expert panel review that information. So they were blinded. They had never seen the patients. They hadn't seen the pathology. They just looked at that information and they said, what disease do we think this patient has? And so we had a large number that had Parkinson's and had dementia with Lewy bodies and two other rare diseases called multiple system atrophy and pure autonomic failure. Then at the same time that the expert panel was reviewing that information, we took the skin biopsies from those patients and we looked to see whether or not we could detect this abnormal protein. So what we found was that in greater than 95% of the patients that were diagnosed as having one of these four conditions, we were able to see that abnormal protein. We call that the sensitivity. So how likely, if you have the disease, can we detect the protein? And the answer was 95.5% of the time. Then the other term that's used a lot in medicine or in science is called specificity. So specificity means how often, if we find that abnormal protein, is the person likely to have a disease? And so the way that we look for that is we take healthy people, people that we don't think has the disease, and we analyze and we look at the same protein. And again, we do that blinded. So when I was reading the pathology, I wouldn't know, what piece of tissue this came from and what disease it came from or whether it came from a healthy person. So in that case, we only found the abnormal protein in 3% of the healthy population. Now, it's likely that some of those people actually had some early symptoms. So we may be detecting people very early in that 3%. There was a fascinating study that was just published out of Finland, where they looked at people that had died out of the hospital, and they looked for this abnormal protein actually in their brains. So they could do an autopsy and they could look for this protein. And these were people that had no symptoms of Parkinson's disease. And they found that you could see the protein about 9% of the time. So probably is a small percent of people that are going about their normal day who have no symptoms, but do actually have this protein already. And that's what we were able to detect. But again, that was only in 3% of the patients. So that meant that our specificity was 96.7%. That when we found the protein, it was associated with one of these four diseases.
That is fantastic. I think it's very interesting to think about that 3% number, because one of the things we work on at the foundation is how do you manage the condition? And we know that earlier you let people know about the condition, the earlier they are able to adopt behavior changes, lifestyle changes that can support them. So being able to improve that diagnosis, make that diagnosis earlier would be so beneficial to patients. And as we think about the study that came out, the high specificity, the high sensitivity, what does that mean for somebody who wants to get this test?
What it means is they can have a lot of confidence in the result. There are a lot of blood tests and other types of tests that are done in medicine where a very high percent of people that don't have the disease may have an abnormal result. And that can lead to, obviously, anxiety. You get concerned that you might have that disease. It can also lead to more medical tests and more doctor visits and, trying to sort out what that means. So we call that a false positive result. The fact that only 3% of the healthy patients had this protein means a very low false positivity rate. Your other point, which is a great point, is how early can we detect this? And we're in the midst of 2 additional NIH-funded studies to try to answer that question. So in this study, as I mentioned, we took patients that we were really certain had the disease. And the reason we did that is that we wanted that to sort of be our gold standard. But we know that that means that patients were a little later in their disease course. So now we're looking at patients that have two other conditions that we know often can lead to one of these diseases. So one is called mild cognitive impairment. And we're looking at those patients to say, can we detect this abnormal protein? And does this lead to dementia with Lewy bodies? And then another condition that I think probably many of the people listening might have heard of is one called REM behavior disorder, which is a sleep disorder that has a very high risk of turning into Parkinson's or dementia with Lewy bodies. And so we're doing another study in patients that have REM behavior disorder to say, can we detect it there? That's a fascinating study because in those patients, they may be having those symptoms 5, 10, or 15 years before they develop Parkinson's. And at least preliminarily in other people's work, our technique, the SYN1 test technique, is able to detect that protein very often in those patients, even years before their disease. So we are hopeful that once we have that data, we'll really be able to prove that we can see these changes early. And to your point, then that means you can start to make those lifestyle changes long before you've lost, so many neurons in the brain.
We've heard from so many patients, including our founder, who after that diagnosis of Parkinson's can go back 5, 10, 15 years and pinpoint the day that they started seeing some of the symptoms and just didn't realize what it was. So I think backing up, making that diagnosis earlier would really help people. And when you touched on about the anxiety, we also hear from people about who call because they've been told it could be one disease or another disease, and they're just kind of waiting to see what the clinical outcome looks like in 4 months, 5 months, 6 months. And of course, the anxiety of that waiting period can be really, really terrifying for a lot of people. So how does somebody get the SYN1 test today?
So it starts by going to see their healthcare provider, the doctor, or nurse practitioner, physician assistant, and really talking about their symptoms. So again, especially in Parkinson's disease, there are many patients who have all the clinical features of Parkinson's disease. They're put on dopamine replacement, they get better. In those patients, there may be no need for a test. The doctor's probably made the right diagnosis, they've treated you with the right medication, and the test is not going to add a lot of information to that. Where we feel we are helpful is when, as you said, maybe it's the beginning of the disease and they don't have all of the clinical features of the disease to make the diagnosis. Or maybe it looks like a different disease and the doctor's been trying to treat and the medications aren't working the way that we expect them to. So it's really useful in that kind of diagnostic dilemma stage where the doctor is trying to say, I want to know a little bit better what's going on with my patient. And then more importantly, also more objectively. So again, rather than just relying on a physical exam, you can now have a piece of information that says, yes, this protein is building up. Yes, I have these symptoms. Therefore, it's likely to be Parkinson's disease or vice versa. I was talking yesterday with a reporter. My father at 89 began to walk hunched over and slowed down. And my mother was concerned that he had Parkinson's disease. And I took him to a neurologist who said, well, it's hard to tell. He's got some other things going on. And we did the SYN1 test on my father, and it was normal, which was very reassuring because then both the doctor and my father was able to say, yeah, this isn't Parkinson's. And now 3 years later, that turned out to be the right answer. So that was really fantastic. So it starts by going to see the healthcare provider, talking about the symptoms that you have. And then if the possibility of a synuclein disorder, a synucleinopathy, is in the differential diagnosis, then the test can be ordered. It's done in the doctor's office. It's very simple. It takes about 15 minutes. It's 3 small little punch biopsies. So we numb up the area, which does burn a little bit with the lidocaine for about 5 seconds. After that, there's no pain whatsoever. There's no stitches. It just heals up like a little scab. And it's about a quarter the size of a pencil eraser. So it's a very small little circle that we take. It just kind of heals up normally.
That's fantastic. You know, how interesting that it hit close to home for you and with your own father and that it was your test that helped to figure that out for him. Do you have any other examples of how clinicians have used this to improve patient care?
Yeah, so we've done a large number of tests across the country. So we've done about 20,000 tests already. So we've helped hopefully,
Wow.
tens of thousands of patients get their diagnosis. For my own patients, I've had some really interesting examples. So I had seen a guy who was, let's say, but roughly early 60s, so relatively young, healthy guy. And over a period of about a year, he started to say that his legs felt weak and he just couldn't walk anymore. And it got to the point where he couldn't go to the grocery store with his wife because he couldn't feel like he could walk through the grocery store. But he kept describing it as weakness. And so when he went to see the doctors and the neurologists, they did all the tests that you would do if somebody was weak, to look for a muscle disease or to look for a nerve disease or a spinal cord disease. And so he had many, many tests and nobody could come up with an answer. I was fortunate because I saw him a little bit later in that whole story. And by the time he got to me, he looked very slow. And I wasn't convinced so much that it was weakness as it was slowness and bradykinesia. And so I said, why don't we do the SIN1 test? The SIN1 test came back abnormal. I felt he had early Parkinson's and put him on Cinemet. And he called me like 2 weeks later, just the happiest man he could be, because he's like, I can walk again. I can keep up with my wife and I can go to the grocery store. And probably if we had waited another year, it would have been clear that it was Parkinson's. But to get him onto the right medicine earlier and to get him functional again was, he was just thrilled with it. So we have lots of those kinds of stories where, again, the whole clinical picture isn't as evident, but some of the symptoms are consistent and the test then pushes us in the right direction.
Yeah, well, we hear from people who tell us all the time when they finally get the diagnosis and they finally get the medications, it's like a light switch goes on, everything changes. And so to move that forward faster, I can imagine is really helpful and really beneficial. So beyond patient care, do you see other uses for the SIN1 test?
So I think one of the big uses is really in the research space. And so we are involved in, I think at this point, about 30 different clinical trials. Some of these are big NIH-sponsored trials. Some of them are very small, what we call investigator-initiated trials, where a doctor, for example, at Virginia Commonwealth University has an idea where this test might be helpful, and we do a little research project with him with the idea that eventually it becomes a bigger research project if it's successful. But probably the most exciting work that we do is with the pharmaceutical companies. And we have, I think, two very important roles to play with the pharmaceutical trials. The first is that we know that there is a significant misdiagnosis rate of Parkinson's disease. So if you take people who are diagnosed with Parkinson's during their lifetime, and then you do autopsies when they pass away, the neurologists are wrong about 20 to 30% of the time.
Wow.
So if you're trying to do a research trial in Parkinson's disease and you're putting 20% of the patients into that trial that don't have Parkinson's disease, it's going to be very difficult for that drug to ever be successful, right? So we talk about that as being patient homogeneity, making sure that the patients you put into a trial are the right types of patients. The second way that we're helpful with pharmaceutical trials is because we can visualize the protein. And because the current thinking, at least, is that if you could lower the level of that bad protein, the disease would get better. You have to have a way to measure that protein. And we just launched a system that we call Nerveillance, which is a computer-assisted device, a software program that allows us to quantify the amount of the abnormal protein. So the pharmaceutical companies are very excited about that because there has never been a way to do that in living people.
Yes. Yeah.
So if you could start a person on a trial and then repeat the skin biopsy at 6 months or maybe 12 months and show that you've been able to decrease that protein, that would lead to FDA approval of a drug. And we saw that with Alzheimer's disease. So the Alzheimer's drugs that have been approved in the last couple of years, one of the main reasons they got approved was because they could show that they lowered the level of the amyloid, which is the bad protein that builds up in Alzheimer's disease. So the pharmaceutical companies have been very excited about our technology because if they have a drug that can lower the level of the protein, we may be able to prove that. So we're hopeful that we can actually speed up these clinical trials and get real disease modifying drugs for Parkinson's to patients much faster.
That is really exciting. And it does paint a very hopeful future for Parkinson's research. And I'm sure everybody listening will be very excited to hear that news. It does make me wonder a little bit about past trials, drugs that may have failed in a trial because we just simply didn't have a way to test, or there just wasn't ever homogeneous population, like you said. Is there any way to go back?
Probably not. But I think it's very likely that there may have been drugs that could have worked, and a couple of the drugs that were very close to working. But again, if you've got these patients in the trial that don't belong there, it's going to wash out the effect, and you won't hit the statistical significance that the FDA requires.
Yeah, because I do remember a couple of hopeful drugs in the last decade, more, and then the huge disappointment as they move through the phases. So exciting news for the research community as well. So, one of the other things you touched on a little bit about earlier diagnosis, we hear from a lot of people from underrepresented groups who, as we know, are often later in getting a diagnosis, maybe not referred to specialty care. How do you see the SIN1 test helping with a diagnosis and access for those groups?
Well, so right now, for example, or before the SIN1 test, one of the main tests to kind of help with the diagnosis of Parkinson's was an imaging test called a DAT scan. DAT scans certainly have their role in the diagnosis. They turn out to be a little less specific than the SIN1 test. So there are many diseases that can cause an abnormal DAT scan. Not all of them are Parkinson's disease. So that's one limitation. But access, for example, to a DAT scan is very limited. So it has to be a nuclear medicine facility. It's sometimes difficult to get the medicine that you have to inject to be able to do the X-ray in the first place. And there's not a lot of these facilities across the country. So access to the kind of ancillary testing that we used to do was very limited. The other point is that the SIN1 test can be done by general practitioners. So if people are in rural areas where access to a neurologist is difficult, and let's say a doctor is trying to decide between is this an essential tremor or is this a tremor of Parkinson's disease, a general practitioner can do the test as well as a neurologist can, probably better than, truthfully, because they do it as part of normal practice and can get the test done. So the fact that it's done in a doctor's office can be done, in the middle of Iowa or the middle of South Dakota. You don't have to drive for hours to get to one of these facilities. I think will really make access much easier for patients.
100%. I mean, we're in Portland, Oregon, based in Portland, Oregon. So we have many, many wonderful Parkinson's programs here, but I know we get people who drive from the other side of the state, sometimes outside the state, to get here, and to know that they can have that tested at a doctor's office at just their primary care. That will improve a lot of access. So, just one last thing. Can I ask what's next for SIN1 test or CND Life Sciences?
So we have a lot of research and development that we're still working on. I think one of the big issues is how does this quantification of the protein relate to the stages of a patient's disease? So in the JAMA paper that just came out last week, one of the things we showed is that the amount of the protein correlates with disease severity. So as the disease progresses, a person builds up more of the protein. And that kind of lends itself to the pharmaceutical trials we talked about, trying to lower the protein. So we'd like to understand a little more about what that quantification means and maybe try to bring that into clinical practice. Another area that we're very interested in actually is other places that you might see the protein. And one of the fascinating places for a disease like Parkinson's, which everybody considers to be a brain disease, is that one of the first places, both in animals and humans, that the protein starts to accumulate is actually in the gut. And so if you look at people that have constipation, they have a much higher risk of developing Parkinson's disease, which is this whole gut-brain axis, which we pay a lot of attention to these days. So we're actually involved in a number of studies now to ask the question, could you actually see this, for example, on a routine colonoscopy?
Wow.
right? So imagine now everybody at 50 or 45, whatever it is, goes for their routine colonoscopy. You could get one extra biopsy and say, hey, are you building up synuclein? And then if you were, imagine now that we have drugs to slow down the accumulation of the protein, you might actually be preventing Parkinson's disease as opposed to trying to treat Parkinson's disease. And that would be really, really exciting for us. So we're very interested in the gut right now as well.
That is really exciting. What a hopeful note to end on. I have to say, a lot happening right now in the gut and the microbiome research. I know that whenever we do any kind of educational programs around the microbiome, they get so much attention. So I think it's fascinating to think about how SIN1 test and what you're doing at CND Life Sciences can help support that work. So Dr. Levine, thank you so much for joining us today. I am really grateful to have a chance to speak with you, and good luck with all your future endeavors.
Thank you. I've really enjoyed this, and have a great day.
You too. Thank you.
The Brian Grant Foundation empowers people impacted by Parkinson's to lead active and fulfilling lives. Help support our efforts at briangrant.org. A special thank you to Raphael Sadiq for providing the music for this podcast. This podcast is produced by Brian Griggs. Learn more at griggsproductions.com.